All right, everyone, welcome to day 2 of cellular respiration. Last notes were a little tricky. They were kind of difficult, I would say. Probably one of the more challenging sections that we will cover throughout the entire year. But if you watch the lecture again or cover the material again, I think you could get a good grasp of it. If you haven't done that already, I definitely encourage you to find another source or rewatch that video to really make sure that you have all the steps down for cellular respiration. Last time we kind of left on a cliffhanger talking about what happens if we do not have oxygen present. We want to remember that last time in class when we did talk about cellular respiration in the presence of oxygen, we covered all of these steps starting with glycolysis. Glycolysis occurs in the cytosol and it actually doesn't need oxygen. Glycolysis will occur in the presence of oxygen or without it. So that's always going to happen. But if we do have oxygen, all of these steps over here are going to occur in the mitochondria. We've got pyruvate oxidation, which is the first immediate step right before the citric acid cycle. So I consider those two things kind of closely related, but we want to make a distinction between like what is pyruvate oxidation versus what is the citric acid cycle. And then finally, deeper into the mitochondria, we have oxidative phosphorylation, which is, of course, the electron transport chain and chemiosmosis. From all of that, we get a total of around 32 ATP. But again, today's focus for class is talking about what happens if we do not have oxygen. So cellular respiration without oxygen. Okay, so we want to remember that in cellular respiration, oxygen is the final electron acceptor. It drives electrons down the electron transport chain. So how do organisms produce ATP in the absence of oxygen? There's two major ways we see this happen. We see that in anaerobic respiration. So these are going to be organisms that would not thrive in an oxygen-rich environment. Oftentimes we find these organisms in environments that are depleted of oxygen, that have very little oxygen. So like bacteria that are anaerobic will undergo anaerobic respiration. And we also see fermentation. This is another example of where we would not have oxygen. Very closely related topics or processes, but we will talk about the differences between these two, anaerobic respiration and fermentation. The first one, anaerobic respiration, generates ATP using the electron transport chain in the absence of oxygen. So this takes place in, again, prokaryotic organisms that live in environments with no oxygen, anaerobic bacteria. And the final electron acceptors here are going to be sulfates or nitrates. So you're going to find lots of sulfur and a lot of nitrogen rich environments. And these are where these anaerobic respiration or these anaerobic prokaryotes will undergo respiration and be able to produce ATP. Now, they don't need to make a lot of ATP. If you remember from last class, how many ATP do we generate after glycolysis? Well, we generate only 2 ATP total. So they're not generating a lot of ATP, but they are generating an energy source best way they can in these types of environments. Fermentation is a second type of respiration without oxygen, and this is going to generate ATP without an electron transport chain. It's an extension of glycolysis. And if you also remember from glycolysis from last time, one of the products of it was NADH, and we were able to move that NADH all the way to the end to the electron transport chain. In fermentation, we don't do that. We do make NADH, but we need to actually recycle it. So then we recycle that NADH back into NAD+. This step does occur in the cytosol. It does not take place in the mitochondria. No oxygen present. And there's two types. We see alcohol fermentation and lactic acid fermentation. So let's differentiate between these two, alcohol fermentation and lactic acid fermentation. Alcohol fermentation, here are the big steps. We have glycolysis, where we take glucose, and we're still going to break it down into 2 pyruvates. And this is, again, a very simplified flowchart of how this all works. We get 2 net ATP. Remember, there's an energy investment phase and an energy payoff phase. We need to invest 2 ATP earlier on, but we get 4 ATP out. So that means we get a 2 net ATP gain. In this whole process, we still make NADH. So I would say from this bracket over here showing us glycolysis, this is still the same as what we talked about last time. Now, the big thing that's going to be different is how we treat this pyruvate. What do we do with this pyruvate, and what do we do with this NADH that we've made? The pyruvate is actually going to be treated as a byproduct, and actually need to get rid of it. So we're not going to go through pyruvate oxidation, and we're not going to go through the Krebs cycle. So we have no use of this pyruvate right now because, again, we don't have any oxygen present. And the NADH, we need to get rid of that as well, because if we don't have oxygen, then we can't do the electron transport chain, and we can't do any of the chemiosmosis that we saw. So we need to get rid of these two things, but we can do that in the same step. So we take those two pyruvates, and then we are able to get rid of only two of those carbons, from one carbon from each pyruvate. So then we end up with a carbon dioxide, and we're using the oxygen from the pyruvate. So like, we're not getting oxygen from the atmosphere. We're getting oxygen from the pyruvate and combining with carbon to form CO2. So one pyruvate will make one CO2, another pyruvate will make another CO2. So now we're down to 4 carbons. We convert those into acetaldehyde, and then acetaldehyde is then converted into ethanol, and that process actually reduces NADH back into NAD+, and then we can use the NAD+ back over here into this step. So in this whole process, when we get rid of the pyruvate, we're turning it into ethanol. Now, a lot of times, people that are 21 and over use this to their benefit because ethanol is the major alcohol that is used as a drinking alcohol. So this is how this whole process is done. You have to ferment using yeast and some other form of a carbohydrate. Like, it's either going to be grapes, or it's going to be something with wheat or barley, like if you have a beer. Those things require oxygen depleted environment. So you put yeast into a liquid, and then you seal the liquid, you seal the bottle with a cork, and you deprive that whole environment from oxygen, forcing the yeast to undergo alcohol fermentation and producing ethanol. All right. The other form of fermentation that we see, and this is going to be more common in animals, is going to be lactic acid fermentation. And muscle cells do this all the time. And muscle cells run out of oxygen pretty often when you undergo strenuous exercise, especially if you do something that where you have fast twitch muscles, like sprinters, for instance. They are going to be depleting their oxygen reserves very quickly. In fact, in a matter of seconds, they can deplete their oxygen reserves. So without oxygen, the cells will undergo lactic acid fermentation in order to produce ATP. If you ever talk to someone who's on a track team and runs short distance sprints, someone who typically runs like 100 or 200 meters, and if you ask them what's the hardest race, they'll probably tell you like the 400 meters is probably really difficult for them. And that is because by the time they get to the 350 meter mark or so, they have depleted all of the oxygen in their leg muscles. And so they are doing lactic acid fermentation, and they are not generating the same level of ATP they were as when they were doing normal cellular respiration. Okay. One final note that I want to make is sometimes you'll see the word lactate instead of lactic acid in a problem. And just like with pyruvate and pyruvic acid, or citrate and citric acid, it's the same naming convention. Lactate is also commonly referred to as lactic acid. So just keep that in mind. Now, anyone that has ever performed an anaerobic exercise, such as sprinting or weightlifting, and you've gotten lactic acid in your muscles, you know exactly how that feels. It's going to feel extremely sore, and that feeling can last for several days. There are things that you can do to help resolve that, like stretching, or like a foam roller, or even just like taking a hot shower. It will help release some of that lactic acid from your muscles. And what happens to that lactic acid? Where does it go? Well, we break that down into basically back down into glucose, and that occurs in the liver. Okay, but there is a limit to how fast the liver can do this, and so sometimes you can have dangerous levels of lactic acid that build up in your blood. So if lactic acid builds up and is unable to be broken down fast enough, it can actually lead to a thing called lactic acidosis. And lactic acid, again, it's an acid, and it gets stored in the blood, or gets transported in the blood. And if you have too much of this acid in your blood, it will actually decrease your blood pH to a dangerous point. Every single year during major marathons like the Boston Marathon, or the New York Marathon, or the Chicago Marathon, there are people that sign up for those races that have never trained extensively to run something more than like a 5K. They're definitely not capable of running a marathon. And every single year, there are people that suffer lactic acidosis because they strain their muscles to the point where they're making so much lactic acid, and that lactic acid is building up in their blood. And they could have potentially fatal consequences of building up too much lactic acid in their blood. And that can be quite scary for those individuals. And this is potentially why lactic acid evolutionarily hurts in your muscles. It kind of tells you, hey, you need to slow down. You need to stop exercising so much. The muscles are getting sore. And that's basically your signal to stop doing whatever exercise you are doing, because it can lead to acidosis. All right. Now, lactic acid fermentation, the whole pathway looks like this, where pyruvate, again, it's the same step as before. We still have glycolysis. Pyruvate needs to get reduced into something, and instead of acetaldehyde into ethanol, we just convert it straight into lactate, and we are regenerating that NAD plus again. So very similar to what we saw with ethanol fermentation, but instead now we're making lactate or lactic acid. So here's the whole flow chart. In this whole process with glycolysis and cellular respiration in the presence of oxygen, we're generating 32 ATP, but without oxygen, we have to go through fermentation, and we're only making 2 ATP per glucose molecule. So that's really inefficient. 2 ATP is not a lot, but the reason why we do this is because basically in times where your cells need energy, this is the only energy source possible. So 2 is still better than 0. But it's not great. So this is why it's really important for us to have adequate oxygen when exercising or doing aerobic exercise. Otherwise, you are straining your muscles to the point where you're producing lactic acid, which could be a good thing. Like if you're doing exercises to the point where you are trying to build up muscles, then yes, doing something like no oxygen, anaerobic exercises could be good for that. But to a certain degree, you don't want to overexert yourself. You don't want to run a marathon if you've never run before in your life. So there are instances where you want to be careful of that. The other note that I want to make here is that it's a good thing evolutionarily that we use lactic acid fermentation and not alcohol fermentation in our muscles, because it'd be kind of funny, I guess. You can kind of think about it that like every time you worked out really hard, or if you ran a 400-meter race, instead of lactic acid, you would end up having ethanol in your bloodstream floating around. And the more you exerted yourself, the more ethanol you would have in your blood. And basically, the drunker you would get. So it's pretty good that we're not actually doing alcohol fermentation in our muscles, and we're actually doing lactic acid fermentation. But this concludes the notes for cellular respiration and for Unit 3. Look out for any assignments coming up. Next up in this class, what I would like for you to do is work on a foldable for cellular respiration. It's a really good graphic organizer to help you remember all of the steps. There are a lot of steps involved, so just make sure you put in a lot of detail into your foldable, and usually you would want to add color to help differentiate the different steps.